In our previous segment, we have discussed about the sandwich model which was proposed by Danieli and Davison and it was rejected. So, the next model of the plasma membrane that we have to discuss in this segment is unit membrane model, which was proposed by Robertson in the year 1959. Name of the scientist, year of the discovery, as well as what is the name of the model is very important. So, unit membrane model by Robertson, it was proposed by Robertson in the year 1959, and this is also called as unit membrane hypothesis. Hypothesis means they have deduced the point based on the structures, based on the study, and that is also not a sure kind of thing, right? So, unit membrane hypothesis, what they have basically said regarding the structure of the plasma membrane is more or less similar to the sandwich model proposed by Danieli and Davison. So, phospholipids are basically, first of all, composition. So, plasma membrane is protein and phospholipid bilayer. Plasma membrane is protein and phospholipid bilayer. That means plasma membrane is made up of P to Pl, that is protein to phospholipid. Then phospholipid, it is again amphipathic like that of the sandwich model. So Pl phospholipids are amphipathic in nature, that is they have the hydrophilic as well as hydrophobic and it's present within the same structure. Arrangement too of these phospholipids are same as that of the previous model, that was the sandwich model proposed by Danieli and Davison. So arrangement of phospholipid is same as that of sandwich model. And how was the arrangement? This is the polar head. Towards the head, the phosphate group, the phosphorus group is attached, and this is attached to the tail structure. This is the tail, this is the polar head, and this is the phosphate or the phosphorus molecule attached. That is what we call it as phospholipid bilayer arrangement. More or less, as I said now, that the structure, the points that they also deduced, that Robertson deduced in the year 1959, was based on the sandwich model only, with little modification, little difference. The difference was only one. Sandwich model, which was proposed by Danieli and Davison, has said that the proteins in the structure of the plasma membrane are of alpha globular type. According to the Robertson, in the unit membrane model, plasma membrane contains proteins, but the proteins are beta fibrous proteins. Now they are not globular, they are not spherical, they are thread-like fibrous. That means the number of proteins will increase corresponding to the number of Pl phospholipids present within the plasma membrane. So the difference is that proteins are beta fibrous proteins or beta fibrous type. They have also given the thickness of the plasma membrane, that is between 75 to 100 angstrom. Thickness of the PM plasma membrane is between 75 to 100 angstrom. Other than that, the details are almost same as compared to or as that of the sandwich model proposed by Danieli and Davison. Now let us check based on the points that unit membrane model says by the Robertson. Let us say or let us see that whether this model we have to accept or we have to reject. So the three properties that we discussed there, same three properties we will be discussing in the unit membrane model. So it is checking the model, that is we have to check the unit membrane model is correct, appropriate or not, right, or that suits or fit within the three important properties of the plasma membrane or the first property, or let us draw the diagram. Now the proteins will change, so the diagram will look like this. towards each other. Proteins are on the either side. Head, tail. Now the proteins are fibrous proteins. That means they are not globular. So fibrous protein will be something like this. So in all over the head I am drawing the 4 fibrous protein, because again we have to calculate the P to PL ratio, that is protein to phospholipid ratio, the second point. So the first point, that a membrane must allow any fat or water soluble molecule to come across, to enter the cell. Again, when the fat molecule is coming inside the cell, no issue, because the membrane itself is made up of bilipid layer, that is fat. So fat soluble substance can easily enter the plasma membrane, and from the plasma membrane it will go to the cell. Okay? So right, what about this water soluble substance? Again, water soluble substance will not be transferred. The same logic. Water soluble substance will not be transferred. So I am putting it across. Second, it should have the variable P to PL ratio. Now let us see P to PL ratio. Right? So protein to phospholipid ratios. Again, let us see only this region of the membrane. 1, 2, 3, 4. 4 phospholipid, corresponding to 4 phospholipids there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. There are 16 proteins which are beta fibrous proteins. They are not globular, now they are fibrous. Although the protein to phospholipid ratio is variable, but they could not explain anything regarding the variable thing of the protein to PL ratio. So that means they could not explain, not given any information regarding the movement. No, not given any information regarding the movement of molecules, right? Say for example, if any molecule has to move in or go out, this P to PL ratio will definitely change. And if this is changed, then what about the dynamic nature of the membrane model? That also was not explained, right? So dynamic nature was not at all explained, right? So according to the points that they have given, we are not supposed to accept this model, because water soluble molecules are not entering the cell at all, and we require H2O for the normal life processes, right? Dynamic nature was also not explained. P to PL ratio, variable number, movement of the molecules, was also not explained by this model. So of course we have to reject this model also, right? Now let us see the other details of this membrane or this model. The membrane is basically called as lamellar membrane. So this membrane is basically called as lamellar membrane. Why it is called as a lamellar membrane? Because it has 3 layers. It has 3 layers. So it is also called as trilamellar membrane model. Trilamellar membrane. Now how trilamellar membrane? The 2 protein and 1 entire thing is counted as 1. So it is basically 2 protein layer and plus 1 layer of bilipid layer is present, right? So now again we have to reject this thing also. Again, the 2 proteins that are present will give the dark appearance when it is observed under the microscope. So the proteins layer are basically dark layers, and the bilipid layer is what we call it as the light layer, right? So this membrane model, that is unit membrane model proposed by Robertson, was also rejected. Now in the next part we will see the final